Anirban Mukherjee, and I will be talking about lac operon today. So, what is an operon? Now, it can be stated as a cluster of genes which is under control of a single operator region, or it may also contain some additional regulatory genes also. So to put it simply, we can just say it's a cluster of genes controlled by an operator. And what is the need of operon actually? So think of it in this way: if there is no one in a single room, but still the lights are on, so that's a wastage of electricity, right? So similar in case of organisms or in case of cells, that they need to conserve their energy wherever and whenever they are required. So operon it actually does the same thing. It saves energy. It regulates the production of proteins to the point of time only when it is required, and not before or not after that. So it limits the production of proteins to only when it is required. Now, what is lac operon? Now, already we have discussed what is an operon. It's a cluster of genes. Need of operon, and lac operon is specifically the lac stands for lactose. It is just that lactose, this operon actually promotes the uptake of lactose from medium and its breakdown into galactose and glucose, and ultimately promoting metabolism. So here lactose, it can act as an inducer to the operon itself. So it is an inducible operon. An inducer is the lactose itself, or more appropriately if we state it, it's the allolactose. Moving on to the lac operon structure of E. coli. So we get to see a cluster of genes. So here are the cluster of genes. We see lac I plus, lac O plus, lac Z plus, lac Y plus, and lac E plus. Now this signifies that this is the wild type. Okay. So lac I plus is one of the regulatory genes, and lac I plus has its own promoter and own terminator regions for its own regulation. And lac Z, Y, and A, these are the lac genes. This actually is a cluster of genes, or it is for which are produced together to give certain proteins. And it is under the control of lac operator or lac O plus, and it has its own promoter for lac genes to promote its transcription, and their own terminator sites. So what do they state? What do they stand for? Lac I stands, it produces or encodes for a repressor protein. Lac O plus, it does not encode for any protein, it's just a sequence, it's an operator region. Lac Z plus encodes for beta galactosidase, and lac Y plus encodes for permease, and A plus encodes for a transvestidase. Let's look at lac I plus. So lac I plus produces the repressor. What is the repressor made of? It contains 4 identical subunits, or 4 identical polypeptides formed of same number of amino acids. They together form the lac repressor. Now lac repressor goes and binds to the lac operator site. Binding to the lac operator site prevents the transcription. Binding to the lac operator site actually prevents the binding of RNA polymerase to the promoter site. Okay, so it inhibits the transcription of lac genes. If we look at it in further details, we get to see this operator region is broken down into 3 parts, that is O3, O1, and O2. The green one is the promoter region. I have maintained the color codings. Okay, and O1 and lac Z is in blue, and O2. So lac repressor, it has a high affinity for O1 and considerably moderate affinity towards O3 and O2. So what it does, it may bind to O1 and O3 at one time. So this will be the structure, O1 and O3, and it may also bind to O1 and O2. Now both of these configurations, or both of these binding sites, they prevent the transcription of lac genes. Okay. Now let's look at lac O. It's just a sequence, it's an operator site to which the repressor binds, and if the repressor does not bind, we get to see the expression of lac genes. Let's talk about the lac Z plus. It encodes for enzyme or a protein which is known as the beta galactosidase. It has 2 functions. It can hydrolyze lactose to produce galactose and glucose, or it can also trigger the formation of allolactose. Now, allolactose is a lac operon inducer. That is, in presence of allolactose, lac operon will be expressed. It will express the genes. Okay. So let's directly go into the diagram. It's a probable diagram of how these reactions occur. Now, this beta galactoside enzyme, it has an active site and also a loop, active site loop. This loop is mobile, or it can move. It can move over to the active site, or it can be in some other places. So if it remains over the active site, it remains in a closed conformation, and if it is placed in some other region, it's open conformation. So in case of an open conformation, which we can see here, the lactose, it binds to the beta galactoside active site. We see that the bond is broken, galactose and glucose is broken, and glucose is released, and galactose is still bound to the enzyme, and further galactose is also released. So we see the hydrolytic action of beta galactoside. Next we will see the allolactose formation. Now again, the basic difference between lactose and allolactose is the galactose and glucose are bound by a 1,4 linkage in case of lactose, but in case of allolactose it is bound by a 1,6 linkage. Now when lactose binds to the active site, we get to see the hydrolytic reaction. The glucose and galactose is broken, but the active site, it comes and it loops over the loop, it comes and closes over the active site. Okay, so here once the loop closes over the active site, it provides a binding site for the glucose itself. Now here conformation changes occur, which triggers the formation of 1,6 linkage between galactose and glucose. Further again, when the loop moves away, the allolactose is released, and allolactose, it again is the inducer for lac operon. Moving on to the lac-Y+, the permease. It encodes for a protein which is known as permease. It actually helps in entry of lactose from outside to inside of the cell, or from periplasm to the cytoplasm of the cell. Now it's a complicated process how it enters. Now let's look at it. We see E1 is protonated, and another proton is taken up in this X site. So now once this X is protonated, we see that the sugar or the lactose, this S is the sugar or lactose, it enters through this channel. And here again, in the next step, we see that the lactose has entered. In the next step, we see that the E1, which already contained one proton, it shifts the proton over, or it donates the proton to the E3, making E1 a deprotonated state. So E1 can now form a salt bridge or a bond with R+ over here. So R+ and E1, when forms a salt bridge, it produces a conformation change and opens up the permease enzyme towards the cytoplasmic end. So after this, lactose and the proton, it is released. Now we get to see a deprotonated E3, a deprotonated E1. Now in the next step, what happens is the already protonated X, which is present, which contains a proton X+, it donates a proton to E1. Now again it has returned to its original state, and again we see that the bond between E1 and R+, which we had seen earlier, it is changed, and R+ and E2- has formed a salt bridge bond, or a salt bridge which closes the cytoplasmic end of this permease enzyme. Next we have the lac-A+. It encodes for a galactoside acetyl transferase. Its function is normal. It prevents entry of non-metabolizable substances, so that the cell or the organism which contains this operon does not intake any harmful substances or any substances which cannot be metabolized. Okay. So permease has a large range of specificity. It is not always specific to lactose. Okay. So once this non-metabolizable substance enters into the cell, the galactoside transacetylase, which has been produced, it binds to that substance and acetylates it. And this acetylated substance is thrown out of the cell. Now once acetylated, this substance cannot re-enter the cell again. So this is a trick which is done by this operon to prevent or to avoid metabolic predicaments and to prevent non-metabolizable substances from entering the cell again. Okay. Now we will see a phenomenon known as the leaky phenotype. Now we know that LAC1 shows constitutive expression. That is, the repressor is produced continuously. The repressor binds to the operator site and prevents the transcription, whereas the RNA polymerase cannot bind to the operator. The transcription is reversed. This one is normal, but in due course of time, we see that the repressor dissociates. And in that time, when one repressor dissociates and another repressor is on its way to bind to this promoter region, we get to see the RNA polymerase binding right away to the operator site and initiating transcription even in absence of lactose. Okay, so ultimately promoting transcription, and some amount of these LAC-Z, LAC-Y, LAC-A, these genes are expressed and proteins are produced. Again, once the RNA polymerase has shifted towards the end of LAC-A and reaching towards the end, the operator site and promoter regions are free, and as we know, repressor is constituted as being expressed in absence of lactose. The repressor again reinstates its position, preventing transcription. This is the leaky phenotype. This is the trick, so that lactose can enter the cell even in times when lactose was previously absent within the cell. Okay. Now let's talk about the state of operon when there is lactose present already in the medium. So if lactose is present in the medium, it is already uptaken by the cell due to the leaky phenotype, which already discussed. Now lactose once entering the cell, it forms allolactose by the action of beta-galactosidase, and allolactose is an inducer, or it binds to the repressor protein, inactivating. So repressor once inactivated, it cannot bind to the operator site. As a result, RNA polymerase binds to the promoter and initiates transcription, so producing mRNA which contains all of these genes, LAC-Z, LAC-Y, and LAC-E. This is specifically known as the polygenic mRNA or a polycistronic mRNA. From here, three different proteins are produced, those proteins which I've already known, beta-galactosidase, permease, transacetide. There is a positive control also. In any state, we always require some kind of control, positive, negative, whatever we need. So lactose is present. Lactose is the sole carbon source in the medium. It is taken up by the cell, and allolactose is produced, and it is bind to the repressor, and repressor has been dissociated from the operator site. Inside the cell, we see the formation of a CAP, that is catabolite activator protein. Along with it, we also see production of CAMP by adenylate cyclase, or the cyclic AMP CAMP. Now this CAP and 4-CAMP molecule, they together form a CAP-CAMP complex. This complex binds to the CAP site. Now this complex is necessary to recruit RNA polymerase. If CAP-CAMP complex is absent, RNA polymerase cannot be recruited to this promoter site. So once CAP-CAP complex binds to the CAP site, it recruits the RNA polymerase and initiates transcription, that is in presence of lactose, when the request is not found. So this RNA polymerase starts its action, and transcription has started, and producing the required genes or required proteins. Sorry, expressing the required genes, producing the required proteins. What happens if there is glucose also in the medium along with lactose? What happens is the cell takes up glucose instead of lactose. Now glucose, obviously glucose is the most widely used source as carbon source for any cell. So glucose once taken in or taken up, it inactivates an enzyme which is known as 3-GLC. Without active 3-GLC, adenylate cyclase cannot perform its action, so it's inactivated. Now once this adenylate cyclase is inactivated, adenylate cyclase cannot produce CAMP or the cyclic AMP. So if CAMP is not formed and the existing CAMP which is present, it undergoes breakdown after a certain time by phosphodiesterase. So there is a shortage of CAMP. If CAMP is not present, or if new CAMP is not being produced, there is a shortage of CAMP. So cap cannot bind to the CAMP, or as the CAMP is absent or it is being broken down, and so cap-CAMP complex is not formed. So we see that the cap-CAMP complex is not formed. So there is insufficient cap-CAMP complex, and which ultimately fails to recruit the RNA polymerase to the LAC promoter itself. Ultimately, we see that the transcription of LAC gene drops significantly even though the repressor are removed by allolactose, even if lactose is present, or even if the lactose is present. So the takeaway message from this catabolite repression is glucose inhibits the formation of cap-CAMP complex, and RNA polymerase cannot bind without cap-CAMP complex to the promoter. Cap-CAMP complex is an absolute necessity for RNA polymerase to bind to the promoter region. Another, the negative control, which we have already seen from the beginning, that is the LAC repressor, which is produced constitutively. It binds to the LAC operator and prevents transcription. So this actually is a negative control in absence of lactose. So this whole negative control is in absence of lactose. Now let's check out some mutations. What happens if these genes or any of these genes are mutated? What if LAC-I or LAC-O is Z? What if some of these are mutated? What happens in partial diploid states? Let's look at these mutations. LAC-I minus, this mutation, it produces a mutant repressor which cannot bind to the operator site. So if repressor cannot bind to the operator site, and also if glucose is absent from the medium and lactose is present, or even if lactose is absent, we see a constitutive expression, right? We see a constitutive expression even if LAC repressor is mutant, or even if lactose is absent, because the RNA polymerase can now freely bind to the promoter site. No one is there to stop it. So lactose, presence or absence of lactose doesn't really matter in case of LAC-I minus. Okay. Now LAC-IS, this is another mutant type. It produces a super repressor. Earlier we have seen that the repressor cannot bind to the LAC operator. Now here we are seeing that the LAC-IS is such a mutant. It produces a certain repressor which binds so strongly to the operator that even allolactose or even presence of lactose cannot dissociate it from the operator site. So it's a super repressor. It keeps on binding to the opus. So lactose fails to act as an inducer. It is an uninductible state. Okay. So once it has bound to the LAC operator site, it cannot be induced further. And in case of a partial diploid state, the mutant is a transdominant. That is, in partial diploid state, we see both of the operons are there. I mean, both operons are there from two different organisms. Yeah, in a partial diploid state. So LAC-IS from one of the operon, it produces the protein. It binds to both of the operons. So none of them can be induced. Okay. In case of LAC-I minus, this one is a tricky mutation. In case of partial diploids, in case of normal strain, even it is seen that it produces faulty proteins. Okay. These polypeptides which are produced, these are itself a faulty. Okay. So if the polypeptides or the subunits are faulty, then the proteins which are produced after their aggregation, they form a faulty protein. In case of partial diploid strain, if the another operon which is present, even if it is a wild type, even if the LAC-I is wild type, we get to see that the LAC or the faulty subunits produced from the LAC-I minus T, it interferes with these subunits to form a faulty protein. So both these proteins are faulty ultimately if LAC-I minus T is present in any one of these operands. So as a result, if a protein is faulty, it cannot bind to the operator sites. So again, we get to see a constitutive expression. LAC-IQ and LAC-SQ, these are actually effects we see due to mutations in the promoter region of LAC-I. Here, the repressor are produced at a higher rate, that is, the transcription rate of the repressor gene has tremendously increased. So let's see in two conditions, that is, in low lactose concentrations and high lactose concentrations. In case of low lactose concentrations, we get to see there are a huge number of repressors. In this case, there are 3 allolactose and 1, 2, 3, 4, 5, 6, 7, 8, 9, 9 repressors. If there are 9 repressors and 3 allolactose, then 3 of them will bind to 3 of the repressors. Still, a huge number of repressors are left. Okay, 1, 2, 3, 4, 5, 6 or 6 about 6 to 7 repressor molecules or repressor proteins will be left. They can bind to the operator. So a huge amount of lactose or allolactose will be required to dissociate these all of these repressors from the operator site. And mind you, these are all being constituted to express. So we will be needing a high lactose concentration. Here we can see that the allolactose concentration is very high, and it is bound to almost all repressors. And there are some free allolactose which might also bind to any repressor which will be produced because it's a constitutively expressing gene LAC-I. So allowing RNA polymerase to bind to the operator site and ultimately permitting the transcription. Again, there is a LAC-OC. This is actually this is a sequence. This does not encode a protein. So the mutation is actually a base pair alteration, and due to this alterations, it becomes unrecognizable for the repressor. Now the repressor is this protein is perfectly normal, perfectly normally producing protein. So LAC-I is wild type. It is producing perfectly normal proteins. This protein cannot recognize the operator region. If it cannot recognize the operator region, it will not bind to it. If it doesn't bind to it, the repressor protein is kind of being useless here. In the presence or absence of the repressor doesn't really matter. But the RNA polymerase can bind to the promoter and initiate transcription. Therefore, we see a constitutive expression even if a LAC operator region is mutated, even if the repressor regions are repressor proteins are off. We see a constitutive expression even in case lactose is absent. So yeah, LAC-OC. So this was all about the mutations and LAC. And these are the papers I've read, which I've read and used it to prepare the slides. So you can go through this. You can actually check the DOI and read those papers yourself. And books I have gone through Russell and Molecular and Cell Biology by Lodish, and I've used these books for preparing these slides. So if you have any further questions, you can comment down below. Thank you.